Welcome to the Science Plus Love Equals Cure podcast, the STXBP1 10-minute monthly update. This is podcast number 22. I'm Charlene Sun-Rigby, president and co-founder of the STXBP1 Foundation. I'm also mom to STXer Juno and her big brother, Luca. Today's topics: STXBP1 Family Summit, the $1 Million Bike Ride, the 2024 Impact Report, Capsida, and updates from the American Society for Gene and Cell Therapy, an interview with Mike Boland. Okay, our first topic, the STXBP1 Family Summit. So our summit is coming up. It's going to take place the 18th through the 20th of July in Westminster, Colorado. We're doing this in collaboration with Children's Hospital of Colorado. Our speaker lineup has been announced. So it's going to sound a little bit like the All-Stars. Ming-Shan Zhui from Baylor, Scott Demarest from Colorado, Ingo Helbig from Children's Hospital of Philadelphia, and Capsida is giving an update on their clinical trial. And there will be other speakers as well. The foundation is also going to present an update on the multiple therapies in development for STXBP1. So don't miss it. There's going to be a lot of time to meet and reunite with other STXBP1 families, in addition to the scientific update sessions, research participation opportunities, and family support sessions. The registration link is on our website, and it also has information on the discounted room block at the hotel. I look forward to seeing you there. Okay, second topic. $1 Million Bike Ride. So this is happening in 2 weeks. So please support our riders. There is also still time to sign up. This year, I am riding for Juno, who will be turning the big 12 in June. Thanks to Lucy and her parents, Aaron and Ben Prosser, for leading this campaign with us. We have raised $900,000 to date over the last 3 years, and over the last several years. And so let's get to $1 million this year. So mark your calendars, Saturday, June 14, raise money and get fit. All right, third topic, STXBP1 Foundation 2024 Impact Report. I wanted to call your attention to this in case you had missed it or haven't read it yet. We published our 2024 Impact Report in April. Over $3 million in funding awarded to date for preclinical research projects, translational studies, and patient studies around the world. And that is in addition to the $900,000 I just talked to you about for the with the $1 Million Bike Ride and Lulu's crew. We also talk about our state experts program that has launched in 21 states in the US. So check out the impact report on our website. All right, our fourth and fifth topics together, Capsida and updates from the American Society for Gene and Cell Therapy meeting. And this is going to be an interview with Mike Boland. So I've invited Mike back onto the podcast. We've had some very exciting updates from Capsida and their gene therapy trial for STXBP1 in the last couple of weeks. This is the first gene therapy trial for STXBP1, and it has been cleared by the FDA to begin the initial trial. This is really a thrilling milestone for the STXBP1 community. The trial is expected to begin recruiting the first patients in Q3. I encourage you to check out our website for Capsida's new letter to the community and keep up with updates by signing up on our contact list. Now, let's welcome Mike Boland to talk about the Capsida news and provide an update from the recent American Society for Gene and Cell Therapy conference. Let's listen in. There's been a lot of news about the American Society for Gene and Cell Therapy meeting, as well as this new Capsida clinical trial for STXBP1. I'm glad we have researcher and parent Mike Boland here to help us break down this news. So Mike is the strategic director at the END Center at University of Pennsylvania and Children's Hospital of Philadelphia. He's also dad to Lucas, his most important role. All right, Mike. Well, thanks for being here.
Thank you, Charlene. It's always a pleasure.
So, Mike, let's start with the American Society for Gene and Cell Therapy, or ASGCT, meeting. Can you remind us what the focus of this meeting is and like who typically attends?
This is an international meeting focused on the development of gene therapies, gene-targeted therapies, and also cell therapies like CAR T therapy for cancer or cell replacement therapies for neurodegenerative disorders. And it's mostly attended by researchers, some clinicians, and in recent years, a lot of biopharma and biotech companies.
Mm. Well, that's exciting. And it seems like an area of growing interest for us from an STXBP1 perspective. So I'm curious what the key themes are that you heard at ASGCT and why were they exciting?
Key themes again this year, similar to last year, were engineered capsids for enhanced delivery of gene therapies to the CNS. But there was also a lot of emphasis on base editing and prime editing, which are adaptations of CRISPR that get around some of the off-target effects that CRISPR-Cas9 systems have. But many of these technologies are being employed for metabolic disorders, because it's easier to target the liver and the blood and regions not in the CNS. I think the most exciting news of this meeting was when uh, Kiran Mushinuru and Becca Ahrens-Nicholas um, talked about their base editing strategy in urea cycle disorder, and essentially it was a first in human, first of a kind base editing gene therapy using lipid nanoparticles targeted to the liver. And they got all of this done within about the span of about 8 months, and were able to treat this little boy, KJ, who was 8 months old, with this gene therapy to correct his urea cycle disorder. It was really astounding accomplishment.
So baby KJ is now famous. And why do you see like what was so this is obviously this is a metabolic disorder, as you said. It's not a brain disorder or a CNS disorder like STXBP1, but why is it exciting for the rare disease community? And how do you see that potentially having implications for other rare diseases?
Yeah, I think, ultimately you can envision a scenario 10 or 15 years from now where we have off-the-shelf base editors that we can use to correct different disorders, primarily metabolic disorders right now, because of the targeting is easier with these lipid nanoparticles. For neurodevelopmental disorders, targeting to the CNS is still an issue, which is why there's been so much effort behind, you know, uh, engineering capsids that can cross the blood-brain barrier and get into the CNS through IV rather than direct brain injection or intrathecal injection.
Can you talk a little bit? So that brings us to capsids or vector. Can you talk a little bit about that? And what is the challenge of getting a medicine or a genetic medicine into the brain? And how is a capsid used to do that, to get to the right places in the brain?
That's an interesting question, and it kind of goes to the route of administration for the different types of gene-targeted therapies that are mediated by AAV, or adeno-associated virus. Um, Traditionally, it was done intrathecally or through MRI-guided intracranial injection. That gets the virus to the region where it is needed the most,
So
more
when you're
more
saying
directly.
So when you're saying intrathecal, you're meaning an injection into the spine,
Right.
or intra ICV is an injection into the brain.
into the brain. Yeah, generally the
Mhm.
lateral ventricle.
Okay.
Um, but a number of companies in recent years have focused on engineering capsids. So the capsid of a virus is kind of the outside of the virus, and it's the protein composition of the virus. And companies have been focused on engineering viral capsids in non-human primates, in monkeys, that to identify different capsid compositions that can cross the blood-brain barrier. So you could use IV injection of the AAV, it gets into the bloodstream and crosses the blood-brain barrier and delivers the virus to the CNS. They've also, one of the problems with IV injection of AAV viruses has generally been they require a higher dosage of virus, and that can lead to liver toxicity and some neuropathy in the dorsal root ganglion cells,
Oh, and that's in the
sensory cells
brain, right?
in the spinal cord. Yes.
And when you say virus, you really mean the outside of the virus, right? These capsids don't they're not like viruses that, make us sick in the when in the winter, things like that.
Yes, and adeno-associated virus. So actually, adeno-associated virus, about 10%, I think about 10% of the population has already had exposure to adeno-associated viruses. It's a very benign virus. It's not like flu or anything, even rhinoviruses that cause cold. It's a very benign virus. And
Mhm.
so that's why this has been focused on as a delivery vehicle for gene-targeted therapies for quite a while now.
I see. I see. So you take the outside of that basically you if you will, you use the bag or the pouch, and and that's really what these researchers have been trying to do is find the optimal bag that will go through the
Yeah.
blood-brain barrier.
Yeah, I always think of viruses kind of like if you think of like a basketball or something.
Mm-hmm.
The outside of the basketball is the capsid, and
Mm-hmm.
it's all the proteins that make up the outside that is the capsid, and on the inside you have the transgene or whatever the actual therapy is that's going to be delivered to the
Mm-hmm.
CNS.
Got it. And in our case, that gene, what's going to be inside of that capsid or AAV is an STXBP1 gene, correct?
Correct.
Got it. Well, can you talk a little bit about because Capsida had a presentation at ASGCT, and so what did they present?
So they presented for the first time a lot of their toxicology studies that they had performed in non-human primates. And this is actually really critical to move the therapy into the clinic. You have to have good toxicology data for demonstration of safety. The toxicology data is not even concerned with efficacy. It's all about safety. And one of the things that the capsid has been engineered to do is to decrease targeting to the liver and that dorsal root ganglion, which we discussed earlier. Now, there's still going to be some virus that goes to the liver and to the DRG, but they showed significant reduction of liver targeting um, and to the DRG. Another thing that they also presented was there was no pathology to any of the organs that they looked at. So from all intensive purposes, from what they presented, the virus looks safe.
That's great. And in terms of how they're proposing, we'll talk about the clinical trial itself in a minute, but they did all of this work in non-human primates, right? And
Correct.
they had some earlier studies in mice. Why do we need to look at these different models?
Yeah, that's a really good question too. I oftentimes you want to look at multiple models because it's easier to demonstrate efficacy in, say, a mouse model. So Ming-Shan Xu's group at Baylor was able to demonstrate that with varying dosages of the virus, they could correct seizures, they could correct learning and memory deficits, motor deficits in STXBP1 haploinsufficient mice. And so that was a really good proof of concept in vivo, in animal proof of concept for the efficacy of the virus. But what's true in mice isn't true in humans, or often not. So and primarily in toxicology. So the pharmacodynamics and kinetics between rodents and non-human primates is quite a bit different, and their reaction to AAV capsids can be very different. So it's important to test the capsid for safety in a large animal model like a non-human primate.
Right, right. Yeah. That makes sense. And I guess at the same time, non-human primates aren't humans. So we also have to do a clinical trial in
Yeah.
people to see.
Yeah,
So
we have to, we're trying to, do as much due diligence to make it as safe as possible. But ultimately, like you said, the real experiment is the human experiment, which is the clinical trial.
yeah. And I wanted to go back a little bit because you were talking about, um, how to give the drug. How does the drug get into somebody's body? And talking about how it's challenging to get through the blood-brain barrier. But it sounds like Capsida, because of this special AAV, they are able to not have to do this intrathecal or this ICV or direct brain type of administration. Yeah. So is it, and that's going to be through an IV?
Yes.
Okay. And
Yep. That will be through IV. And one of the aspects of the capsida data that they presented at ASGCT was an enhanced transduction efficiency across
Mhm.
multiple brain regions. And
Mhm.
so when I say transduction, it means the virus was able to get into neurons and different cell types in the brain across multiple different brain regions. And last year at the STXBP1 Summit, they had presented some NHP, or non-human primate, transduction data from the brain, and it wasn't that impressive. But this year, it's their data looks a lot better. They're seeing levels from 60 to 80% across different regions of the brain, and that's encouraging.
Oh, that's great. And why is that important to see that kind of expression across the brain, like for STXBP1?
So one of the outstanding questions that we don't really have a handle on is how many neurons in the brain do we need to transduce or deliver STXBP1 to completely correct the phenotypes that we see. Ming Shan's work in the mouse model suggests that a low dose of virus isn't going to get us there. We need escalating doses, like a medium or a higher dosage of virus, to
Mhm.
start to see phenotypic rescue. And so they performed similar experiments in the non-human primates using dose equivalents in the
Mhm.
non-human primate to what they saw in the mouse that could correct seizures and learning and memory. And then they tested safety at those higher levels of virus, and they deemed that the virus was safe and didn't cause any overt pathology at the higher dosages.
Got it. And so people talk a lot about gene therapies being one and done.
Mhm.
What's actually happening? So you have this gene, right? This, and you call it a transgene. So
Right.
you're carrying the STXBP1 gene inside of this capsid, and it gets into the brain. And then what happens and why, why do we think that that effect goes on? It's
Yeah.
not, kind of a short effect.
Yeah, it's another good question, Charlene, which I'm used to from you. Um, so you would compare like an AAV mediated gene therapy to like an ASO therapy.
Mhm,
ASO therapies, it's just a synthetic piece of DNA that you would inject into the spinal cord every, say, 4 to 6 to 9 months, maybe.
mhm,
But you have to repeatedly do that because the ASO degrades over time.
mm.
With an AAV mediated gene targeted therapy, the virus gets into the neurons and it can just stay in the neurons. It doesn't integrate into the neuron genome or anything. It stays separate from the normal DNA, our normal DNA, but it just stays in the neuron and it just continually makes STXBP1 seemingly forever.
Got it. Okay. So it basically ends up being like an extra piece of DNA,
That's right.
like our, analogous to our DNA that we were born with,
That's
but
right.
it isn't necessarily, it doesn't become part of that original DNA, but it's then able to basically be the basis for creating at more STXBP1 protein.
That's right.
Yeah.
Yep.
Okay. Oh, that's cool. Well, so l- let's talk about the Capsida clinical trial, because they had an announcement in addition to the presentations that you talked about. And so, can you tell us a little bit about when it's going to start and what this initial, like, they're calling it a phase 1/2A, is going to focus on?
Yeah, so their clinical trial synergy, I think it stands for Syntaxin-Binding Protein Restorative Gene Therapy. It's a tough acronym, kind of like STAR. Um, but it's a phase 1/2A trial that's primarily looking at safety and second efficacy. So the phase 1 aspect will be a dose escalation. So it'll be a small cohort of 3 to 4 patients that receive a medium level of virus, medium dose of virus, and then another cohort of patients that will receive a higher dosage of virus. And then we're just going to test, make sure that is safe, that um, the viral loads that were administered are safe first and foremost. And if they are safe over a period of time, then a second cohort of patients will be enrolled to get another to also get the gene therapy, and that will be tested for efficacy.
Got it. That's great. And I know that all of us as parents and people in the community, the development of therapies, we have such urgency around it, and so there's a lot of excitement around this trial as the first gene therapy trial. But I also am really excited about the landscape
Yeah.
because we really have a growing number of genetic therapies that are under development, and you mentioned ASOs
Hmm.
also. So I think that, I really think about this as the beginning of, the next chapter for us in terms of therapy development. What do you think families should do during this time?
I think families need to stay informed through your podcast, through the family meetings. Also, keep your eye on clinicaltrials.gov, and if you're interested in even just learning more about the clinical trial, that's probably the best place. But also, I think it's important for the community to stay engaged with the STAR study, our clinical trial readiness program, which some of that data that's already been collected through the STAR study will be utilized for this first clinical trial, Capsida's clinical trial. But also, what's really exciting is the European counterpart to the STAR study, ESCO, will be kicking off with a few countries starting to see patients, I believe, next month, and other countries coming online when they're ready. So I think that it's important for the communities in Europe and in America to stay engaged, because for this clinical trial, but also, like you said, future clinical trials, this is just the beginning. There will be more clinical trials in the coming years. And we need data, really solid data on good biomarkers and outcome measures that we can use to test therapeutic safety and efficacy in clinical trials.
Yeah, that's a really great point, Mike. And one of the things that I was very excited to see was that the protocol that is in the Capsida trial really aligns with the STAR and ESCO protocols. And
Yes.
so I was really heartened to know that the data that we're collecting is valuable. You
It
know,
is. It's incredibly valuable, and Capsida has been incredibly collaborative during this process. And we kind of designed this initial trial together based on what we've already learned in the STAR study.
well, Mike, thank you so much for spending some time with us today. And please, as Mike said, um, get updates on the Capsida trial at clinicaltrials.gov, as well as check things out for our summit that is going to happen in July. Thanks, everyone.
Thanks, Charlene.
Wow, a lot of exciting information there. Remember, keep up with updates by joining our contact list on our website. Okay, let's call it a wrap for today, and I will see you next month.